Yeah. So before I introduce, so welcome to the Department of Physiology seminar series. So before I start to introduce our speaker today, I would like to share an exciting news to you guys. Our own Dr. Dang Hugeman is now elected to the National Academy of Science. I don't know whether Dang is here or not, but yeah, congratulations, Dang. This is well-deserved. Yeah. Okay, so it's my great pleasure to introduce today's speaker, Dr. Hao Zhu from the UT Southwestern Children's Research Institute. So Hao was born in China and then came here when he was young. So he graduated, he got his bachelor's degree from Duke University. And so he then got his MD from Harvard Medical School and MIT. So during his medical MD training, he actually did a honors thesis in Dr. Lan Zhang's lab in the Children's Hospital in Harvard. And so he studied hematopoietic and vascular development in zebrafish. And later he did residence and fellowships in medical oncology, and also a postdoc with Dr. George Daly in Harvard. There he studied the function of the oncogene LIM28 LED7 pathway in development, tissue regeneration, and cancer. So in 2012, he was recruited to here UT Southwestern as a CPRIT scholar. So he, in his own lab, he focused on the tissue regeneration and cancer, the relationship with cancer development. So his lab identified genes and the mechanism that regulate tissue regeneration, the regeneration capacity of liver, and how it leads to liver cancer. So through their many innovative studies in their lab, so they identified genetic, epigenetic, metabolism mechanisms underlying the regeneration and cancer. So really provided a significant impact in this field. So he also got quite some awards. So he's the recipient of a Bureau's Welcome Career Award for medical scientist. So he's also a physician, attending physician in the liver cancer clinic here in Parkland. So he also recently got the award for as a Mark Foundation emergent leader in cancer research. Today he's going to tell us more about his work in the regeneration and cancer. If you have questions, you could raise your hand or type your question in the Q&A. We will unmute you, and then you'll have the chance to ask questions. So I'm not sure whether Hao would have a, in the middle of his talk or after one session, maybe we'll have some Q&A session, or we could answer the question at the end. And here you go, Hao, welcome here.
All right, thank you so much, Jiang. I'm very excited to be here with Physiology. I haven't spoken at UT Southwestern in a while, so looking forward to your feedback and happy to answer questions whenever. So here's some conflicts. I wanted to first cover sort of a clinical case that we see commonly in my clinic of a liver disease patient in 2021. So an older gentleman or a middle-aged gentleman, diabetes, overweight, with elevated liver enzymes shown to have fatty liver disease on a biopsy. Not quite cirrhotic, but getting to cirrhosis, and this is a diagnosis of non-alcoholic steatohepatitis. And basically what we do these days is try to exert lifestyle changes to try to reduce weight, reduce diabetes, and see if the liver disease can be slowed down. But ultimately this fails and this leads to cirrhosis. And once you get cirrhotic, what we do is we try to surveil these patients with ultrasounds to make sure they aren't coming out with liver cancers. So a number of years later, he finds that there's a mass, an elevated tumor test called the alpha-fetoprotein, and his HCC, or hepatocellular carcinoma, is surgically resected for a cure. So a number of years later, he recurs, which commonly happens after surgery. And he's outside the transplant criteria. So he comes to my clinic looking for systemic therapy or chemotherapy. We give him kinase inhibitors, combinations of kinase inhibitors and immunotherapy. This works for a while, but ultimately this fails, and he passes away from progressive HCC and a combination of liver failure. So unfortunately, this is becoming a very, very common scenario in the United States and in the Western world, whereas before it was much more common in East Asia, Sub-Saharan Africa. So this is a picture of chronic liver disease. And as you guys know, the liver is famously regenerative. You can cut out almost 80% of the liver and it will grow back within a number of weeks. But if you chronically damage the liver with hepatitis viruses, alcohol, steatohepatitis, this leads to this cirrhotic appearance of the liver, hardening of the liver, making it look and feel like a football. And the cirrhotic liver and the cirrhotic patient really teaches us a whole lot about the functionality of the liver, which is extremely important. These patients come to us with yellowing of the skin, with high bilirubin levels, fluid dysregulation or ascites, and then because of their high portal pressures, they become dilated in their veins, in the esophagus and the rectum. They can bleed and die from this. Furthermore, they're not metabolizing their proteins, so they get encephalopathic and they become very confused, hallucinogenic, and their brain doesn't function. And if that wasn't enough, they then frequently get liver cancer or hepatocellular carcinoma, which is the most common primary form of liver cancer that I see in the clinic. So really we're dealing with cancer as well as these many other diseases. And of course, this is a really complicated process, a lot going on over many decades. And my lab is overall interested in what's going on under the hood. What is happening from a more reductionist standpoint? What are the components here that make a difference in cirrhosis? We're particularly interested in the events of injury, cell death and damage, how the tissue regenerates, and what are adaptive and maladaptive factors in this process. And in particular, over the last number of years, we've been sort of focused on genetic events or somatic mutational events that occur in liver damage and disease. And also the cell biology with different cell types and cell fates in the liver. So with these two variables in hand, we alter these variables to try to understand, do they impact liver cancer formation? Do they increase or decrease liver cancer formation? And how does it relate to the tissue fitness or functionality of the liver? Is it necessarily that if you get reduced function, you get more cancer? If you get increased function, you get less cancer? All of these things are questions that we like to ask. And so today I'll tell you about two overall projects or programs in the lab that we're interested in, the genetic events and the cell biology. So the first story I'll tell you about is something that's been developing over the last 10 years in many different groups. And that is that we have tons of mutations in all of our tissues. Even though we are normal, we don't have cancer, our normal cells may not be entirely wild type. And this image on the left here was a seminal work from Inigo Martin-Carrena in Science, showing that when patients who had plastic surgery of their eyelids, when they sequenced those eyelids, they had tons of mutations in recurrently mutated genes across just 1 centimeter of eyelid tissue. And furthermore, this has been shown in many other tissues, showing that there are recurrent mutations in the esophagus, colon, liver, as I will tell you. And the real question in the field is, what is the meaning of all these mutations? What is the impact of this genetic diversity and heterogeneity? and how does it relate to cancer formation? Do these all mark high-risk cancer cells or patients? And so a number of years ago, we asked this question of the liver, and we thought, well, this is probably the best question to ask in the context of a damaged or cirrhotic liver, because then we can look at clonal expansion as a marker of genetic mutations that may have expanded or contracted. We thought that maybe this would uncover new biology and that new mutations in genes that we might not expect might illuminate unknown pathways in regeneration or tissue homeostasis. And importantly, what does this mean for the beginnings of cancer? So, simplistically, what we did is we partnered with Adam Yope and the liver cancer surgeons here, and we took the adjacent livers from patients who were getting resected for liver cancer. And we very naively took chunks of this tissue and just performed exome sequencing, about 2 to 3 millimeter chunks on each dimension. We did exome sequencing. We took about 60 patients from UT Southwestern, as well as patients from the TCGA, and we looked at a variety of different sexes, levels of fibrosis, whether they had HCC or not, and different etiologies. And so, as you might expect from the introduction that I gave, there were many mutations in these tissues. In just a little tiny piece of tissue, there were an average of 4 to 5 mutations that we could detect using exome sequencing, a median of 1 mutation. And the pattern of mutagenesis in cirrhosis as compared to the pattern of mutagenesis in HCC as detected by the TCGA were very similar. So this suggests that the processes of mutagenesis, the etiologies of mutagenesis were contiguous with cancer development. And an important finding that we had made along with a paper that came a little bit later from Peter Campbell's group was that the more cirrhotic or the more fibrotic patients were, or the livers were, the more mutation burden they had. So the increasing numbers of mutations and the clone sizes of the mutations. This also correlated with the markers of liver damage, namely AST and ALT. So this really gave us confidence that the mutations we were calling were correlated with a clinical feature in these patients. When it came to variant allele frequency, we found that the variant allele frequencies were predominantly under 10%, as you might expect, because we were basically sequencing large heterogeneous chunks of tissue, as opposed to doing very fine laser capture microdissection. And so we knew that a lot of the genetic variation or diversity was going to be under 5%. Now, I'll say that 5 to 10% is a low variant allele frequency, but if you think about it, if you have a 10% variant allele frequency, that suggests that 20% of the cells within that tissue are heterozygous for that mutation. So it is low, but there's still a good number of cells that are mutated here. So we decided to look deeper, and we use ultra deep sequencing, targeted sequencing of about 150 genes, some of which are HCC related, and some of which were identified in the exome sequencing that we had found. And so we repeated this analysis on about the 60 patients. And what we found was that there indeed were a number of mutations that were seen in HCC, like P53, ARID1A, albumin, ApoB, and ARID2. But there were also a large number of mutations that were not commonly seen as recurrent mutations in HCC. And a few that I'll talk a little bit more about, PKD1 and KMT2D, along with PKHD1, which is also a polycystic kidney disease related gene, PPAR-GC1 beta. So clearly there were a number of genes that were recurrently mutated on the gene level that were not necessarily drivers of liver cancer. And we wanted to compare the results with clonal hematopoiesis, which is, of course, clonal hematopoiesis, the blood is flowing through the bloodstream, and it's all, the mutations can be detected with each sampling. With the liver is a solid organ, and the lobules are restrained to geographic locations. So when we tried to do sequencing on different disparate parts of the liver, we found that mutations were rarely shared between different parts of the liver. unless they were contiguous pieces of tissue that we could, that we knew we were sequencing adjacent pieces. So this made sense. And in a way, this indicated that maybe the fibrosis that occurs in cirrhosis may serve as a wall or restriction of clonal expansion that might spread throughout the liver. And I think a key thing that we were trying to do in our study that was a little bit different than other genomic studies was to ask if there are functional impacts of some of these genes in liver homeostasis. And for this, we turned to a project that Joyce Jiang, a graduate student in my lab, was working on, which is to set up an in vivo CRISPR screening approach to evaluate pooled genes or pooled libraries of genes in the context of regenerating livers. And so she had generated these transposon cassettes borrowed from Lars Zender and Scott Lowe's shRNA approaches, where she took guide strands against any gene of interest, Cas9 contained within here, fused to an FAH cDNA, which is a rescue gene for a disease called hereditary tyrosinemia. So this is a metabolic enzyme in the tyrosine pathway, where if you knock this out in a mouse, they get liver failure and hepatocytes die. But if you add this gene back, those clones survive and expand. And so we could give this plasmid in the context of a Sleeping Beauty transposon or transposase, inject it in the tail vein, it will shotgun the liver, and then these will expand to regenerate the liver because they contain the wild type FAH. Then you grind up the liver and you do deep sequencing, and you can figure out which clones expand or contract in a relative difference with control guides. And so this is just to show you that it works. This is a guide strand against PTEN that deletes PTEN on the protein level. This is PTEN IHC. Non-targeting guide does not get rid of PTEN. You do see increased levels of FAH wherever clones are integrated. And then if you give it time, PTEN will be lost across the liver and FAH will be gained across the liver. And what was really neat about this model is that when you look at the PTEN deficient liver, it looks exactly like the PTEN flox allele that was made many, many years ago, where you delete PTEN in the entire liver and you get this fatty liver. So this really suggested that this clonal method of deleting genes was mimicking the more classic flox allele methods that were used previously. So we knew the system worked. And we deleted a number of genes that were found to be mutated at some level within our cirrhosis sequencing. And I will comment here that these are loss of function assays. They're not mimicking the mutations that we found. They're merely trying to identify whether or not these genes have a significant functional effect on hepatic homeostasis. And so on the first couple of iterations of the screen, we found that it worked quite well. And as you can see here, this is a plot examining the repopulated liver compared to the sgRNA reads that we put in initially. And a number of outliers jumped out, including ARID1A, PTEN, and PKD1. And of course, we've talked about PTEN at the clonal expander when you delete it. And ARID1A was quite satisfying because we've been studying ARID1A in regeneration using classic mouse models. And this really revalidated the fact that when you delete ARID1A, it creates a hyperproliferative situation within the liver. PKD1 was unexpected, but interesting considering that it was the top gene in our recurrently mutated gene list. So we focused on a couple of genes that were less well studied in the context of liver regeneration. And one of them, KMT2D, which is of course a important epigenetic regulator, a histone methyltransferase that is mutated in cancer, but also mutated in many normal tissues, including bowel, bladder, and other tissues. So what we did was we decided to focus on heterozygous flox alleles because we thought, well, in humans, it's probably that one allele is mutated and not both in the cirrhosis sequencing results. And so we gave CRE or GFP as a control, and we looked at these KMT2D livers in the context of injuries like carbon tetrachloride or thyroid hormone diet, which causes hyperproliferation. And in this context, in the carbon tetrachloride situation, You get centrilobular necrosis because carbon tetrachloride kills hepatocytes very readily around zone 3 in the centrilobular space. And in the heterozygous mice, this was much reduced. And this is validated by reduced AST and ALT as well. And furthermore, this caused hyperproliferation in the context of thyroid hormone, which suggests that there's a dual effect here. There's a reduction in injury and an increase in proliferation, really suggesting and supporting the idea that the mutations in the human disease have some functional relevance. So this was interesting. And then we also looked at PKD1, and this is a gene that has been quite interesting for many reasons. It's a famous gene involved in autosomal dominant polycystic kidney disease, which many people at UT Southwestern have an interest in. It's a very common monogenic human disease where you have germline deletion or mutation in one copy of polycystin 1, and then there's a second hit that leads to cystic kidneys as shown here, and a need for kidney transplant. This also can lead to cystic liver disease, although this is much less common than cystic kidney disease. Now, the interesting thing is that PKD1, polycystin 1, and polycystin 2, both implicated in this disease, are transmembrane proteins that interact with each other, presumably in the cilia, but it's not clear what the molecular function downstream is. It's been a very vexing field. I'm a newcomer to the field, but it's been very vexing because it's unclear what the mechanisms are. So as I said, present in the cilia, physically interact, features of adhesion, G protein coupled receptors. And it's been observed that when you lose PKD1, this activates oncogenic pathways and cancer metabolism signs and symptoms. And also that there's some more recent evidence that PKD1 is a mechanosensor, that it exerts influences on calcium signaling in response to stressors, mechanical stressors in the environment. So these are things that are kind of known about PKD1. We decided that we really needed to revalidate whether or not our PKD1 mutations that we found in our first cohort were real. So we went to 32 more patients, distinct patients, males and females, different etiologies, but predominantly hepatitis C, and different fibrotic stages, but more advanced fibrosis or stage 4 fibrosis. And this time, Min Zhu in the lab really did a careful job of segregating individual patient livers into 4 or 5 fragments each, including the blood, and looking at the adjacent histology. So we did ultra deep sequencing on all of these fragments. And again, what she found was that PKD1 was the most frequently mutated gene, seen in about 37% of this cohort. So 12 of 32 patients had PKD1 mutation, which is quite remarkable considering we're biopsying one small piece out of this patient. And the variant allele frequencies, although range from very low to reasonable number, 13%, which is about a quarter of the tissue. And moreover, what we found is that multiple fragments had the same PKD1 mutations in a lot of samples, suggesting that the clones were growing beyond just a tiny little piece of liver. Incidentally, we also noted that insulin receptor, ApoB, albumin, PPAR-GC1 beta were again found to be mutated in these samples. And an interesting observation that P53 and beta-catenin are actually quite rare, even though they're the most commonly mutated genes in HCC. So it really suggests that if you see a P53 or beta-catenin mutation, it's really an indicator that you may get cancer from those lesions. So we went on to validate a handful of these PKD1 mutations using Sanger sequencing, along with PKHD1 shown here. But in observation, something that is known in the PKD1 sequencing field is that there are many pseudogenes of PKD1, pseudogenes that can cause problems for exome and ultra-deep sequencing. So what Min decided to do was to really make sure that the mutations we were seeing were in PKD1 and not a pseudogene by performing long-range PCR using PCR primers that are very specific for PKD1. Doing these long-range PCRs and then doing nested PCR around regions we found to be mutated. So using this approach, she found that most of the mutations we found in the ultra deep sequencing could be re-identified using a long-range PCR approach. So this gave us confidence that PKD1 is really actually mutated in a good number of patients in these livers. All right, and again, I'm turning to a functional study that we did with help from the kidney core who had provided the PKD1. It was either Tom Carroll or the kidney core, I don't remember, but PKD1 flox alleles were provided to us, shared to us. And we again did the same kind of experimentation where we injured the livers. We found that there was a reduction of liver injury as seen by a reduced amount of necrosis after carbon tetrachloride. And if we kept giving carbon tetrachloride, this would result in a lower level of fibrosis over time. Now, interestingly, the proliferation was not really different after PKD1 deletion or carbon tetrachloride injection. So it seemed like it was more of an injury response rather than a proliferative response. So given the fact that PKD1 is not really commonly seen in HCC, and we've done the analysis of the TCGA HCC mutations, and we find that there's not that many PKD1 mutations in HCC. Certainly as a fraction of the total population, it's underrepresented as compared to cirrhosis. And so we did the cancer-inducing experiment where we took PKD1 floxed mice and crossed into a liver Cree, and then gave a mutagen at day 35 and waited a year. And this is a very slow model. And what we could see was that there's not a significant difference and actually a trend towards lower numbers of tumors in the PKD1 deficient livers. And what's remarkable is we saw that these livers are very pink and red, but we still don't understand why there's this color difference. Maybe there's something to do with hypoxia or vascularity, but we haven't gotten to the bottom of that yet. And we then did another experiment where we somatically mutated PKD1 after postnatal development at around 3 or 4 weeks, and then repeated the study that I just described. And here we saw a clear result with reduced tumorigenesis in this model. So this functionally really shows that PKD1 deficiency in the liver is associated with suppressed tumorigenesis, although it protects against fibrogenesis. And around the same time that we had gotten these results, a PKD1 expert had gotten in touch with us and said that asked us if we knew that patients with PKD1 deficiency that go on to get kidney transplant actually have a lower rate of cancers in other tissues than do patients who go on to kidney transplant without PKD1 mutations. So it was a really interesting epidemiological finding that is associated with the kind of results that we're getting. Now, we admittedly at this point are having a very difficult time trying to pinpoint the mechanisms. And we've looked at a lot of things, mTOR, PI3 kinase, YAP, Hippo, and I know DJ has looked at PKD1 and Hippo, and we just have not seen the same kind of relationship within the liver. And so transcriptomics has been done, and we're sort of looking at different pathways in this context. But right now, the mechanisms in PKD1 in the liver are still a work in progress. I think at this point we can say that, we believe that there's a whole world of mutations in the liver and many other tissues. And traditionally we've thought of mutations as being a harbinger of cancer and a stepwise progression towards cancer. But we know now that there's a lot going on independent of cancer, independent of liver cancer. And that doesn't mean it's not interesting. There's a lot of interesting physiology going on here. So I think there are many implications of this kind of study. And I think, regeneration fitness promoting mutations may not always be driving and connected with cancer. And the mechanisms for this are going to be really interesting. In other tissues, are there going to be mutations that are influencing function that are independent of cancer? And I think there's already evidence of this in inflammatory bowel disease, where certain mutated pathways appear to reduce inflammation in the local environment rather than increase inflammation or increase cancer. I brought up the point about P53 and beta-catenin. Maybe this suggests that certain mutant clones, if they appear, are really specific for cancer. And finally, I think this kind of physiology, the normal biology that occurs in humans, could really indicate that this kind of assay it could serve as a fitness assay for all kinds of new biology that we might discover. And I'll just give a few comments on that in terms of what we're doing now. And of course, many people here are experts in fatty liver disease. We are not one of those people, but we've become interested in this idea of cell competition in fatty liver disease. And I introduced a patient with fatty liver disease earlier. This is driven by, or at least associated with, obesity and diabetes, as well as underlying genetics. It's extremely common, leads to cirrhosis, especially if inflammation follows steatosis or simple fat accumulation. Furthermore, we predict, and in fact, there are going to be studies showing that there are recurrent somatic mutations that are associated with lipid handling genes within NASH livers. And this really suggests interesting specific biology that correlates with the etiology of disease. And I think if true, this could indicate that NASH could be genetically or modified or reversed within subclones within the liver tissue. And this introduces an idea that maybe cell competition can be used to study fatty liver disease. Cell competition being traditionally defined as there are heterogeneous levels of fitness associated with somatic or epigenetic or genetic variation. Loser cells get taken over by winner cells. Winner cells take over less fit cells and clonally expand. And so we think we can use this approach to understand, is this why somatic mutations might clonally expand in the diseased liver? And I think we can use this to systematically identify genes that change cell fitness in NASH. We can prioritize the most important genes and potentially discover therapeutic targets in this way. So what we're doing now, and we're just getting the results of this, so I don't have much to report in this regard, but we developed a new in vivo screening system, which I think is going to be the next generation approach and very powerful, which is to take Cas9 expressing mice and then deliver AAVs with large pools of guide RNAs. The AAVs will integrate into the genome of the hepatocytes because they carry transposon integration cassettes. So the traditional problem with AAV is that they never integrated, so you could never read out the results of the screen. So with transposon cassettes, we can then read out what is being integrated into that cell. This generates the genetic diversity. We then put these mice on high fat, high sugar water, and then ask what happens in terms of clonal expansion and contraction for a large number of genes. And we compare this situation to normal diets where you don't have this environmental perturbation of metabolic syndrome. So the differences we believe between these two conditions are really going to pinpoint underlying biology that's maybe independent of just cell proliferation. And you might expect that cell fitness will be changed for a variety of reasons. The fitness could be changed because you're knocking out a tumor suppressor. The fitness could be changed because you're preventing lipotoxicity of that hepatocyte clone. So there's going to be all kinds of things that we can imagine here, but the results will tell us if our hypotheses are correct. So hopefully next time I talk, we'll have a whole slew of new biology that we can discuss in this regard. And I want to spend the last half of the talk talking about a project that we've been working on, that a big part of the lab has been working on over the last 3 or 4 years. And that's to sort of systematically understand the source of hepatocytes and heterogeneity within hepatocytes in homeostasis and disease. And I won't go through this, but clearly there's going to be importance in understanding the cell biology of the liver, whether or not there are differences between different hepatocytes and how that might relate to normal homeostatic growth and also disease-related growth. This is going to impact regenerative medicine and cancer biology as well as cirrhosis biology, because the different cells that might expand may have an impact on the ultimate function of the tissue downstream. So when you look at the liver, you guys are probably thinking, well, this is just a sheet of cells. It's all doing the same thing. It's quite boring. And it's a monotonal, sort of monotonous histology. But in fact, the biology is quite interesting. Um, there's an organization in these hepatic globules that are actually sort of hexagonal in shape and football-like in 3 dimensions, or submarine sandwich-like, you could say. And if you cross-section these, they're organized into different zones, zone 1, 2, and 3. And blood flows into the portal triad, down the sinusoids, into the central vein, and back into the heart. So recently, many groups have shown that there's a great amount of diversity in zonation and different gene expression. And up here are zone 1 related genes, down here is zone 3 related genes. These genes are around the central vein, up here are around the portal triads. And in fact, single cell sequencing by several groups, including the Itzkovitz group in Israel, has shown that there's a massive diversity of hepatocytes. If you look at this from a functional standpoint, there's a lot of heterogeneity across the lobule, with high oxygenation blood appearing in the portal vein and getting more and more hypoxic as it goes to the central vein. Energetically demanding functions occurring in zone 1 are commonly seen, and other functions like lipogenesis, ketogenesis, glycolysis occurring around the central vein. One key effector of this is Wnt signaling, and probably oxygenation of the blood is another key effector, but the exact determinants of zonation are not entirely clear. The first step is to ask whether or not there's diversity in function in terms of regenerative and stem cell biology. And a number of groups have nominated stem-like cells occurring across the liver, of which I'll tell you about just a few of them. So Rolnuce's group has shown that a group of cells around the central vein expressing axin 2, which is a Wnt target gene, appear to expand over time, expanding to cover about 30% of the liver from this very central vein, zone 3 position. Another group, Michael Karen's group, has shown that a SOX9 population around the periportal region, this population of hybrid hepatocyte bile duct cells, they don't do anything under normal homeostatic conditions, but if you damage the liver, especially around zone 3 in the central vein, there's a streaming of these hepatocytes toward the central vein because they're proliferating to regenerate the lost tissue. So this is thought to be a stem-like or a progenitor-like cell. And more recently, Steve Artandi's group showing that telomerase high populations distributed across the liver can give rise to new hepatocytes across the lobule under normal and injury conditions. So clearly, this has shown that there's a lot of differences in opinion and differences in observations within the field. And so we really wanted to ask, are there liver stem cells, or do all hepatocytes contribute equally? Is there geographic differences? And so what we did was we, with support from the CRI and the Genome Engineering Corps, we made a whole series of new Cre-ER lines, integrating Cre-ER into the most famously zonated genes within the liver. And so this is actually a much easier technology now than it was maybe 10 years ago because of easy CRISPR and different methods. You can make homology arms around your cassette, and then you can integrate them into the targeted region of interest. We targeted the 3' UTR because this was less likely to cause perturbations in gene dosage, which has been shown to be an important factor in lineage tracing results, because changing the dosage of these genes may affect homeostatic repopulation. So anyways, we generated these lines, and this is just to show you a sampling of some of these, that we could cover the entire hepatocyte population, all the hepatocytes except some around zone 3, and then sort of graded from zone 1 to more and more zone 3, covering different lobular populations. And I'll go through some of these in detail. So we did the simple thing, was just to do a side-by-side comparison of all these populations, zone 1, zone 2, zone 3, as well as bile duct cells. And we give tamoxifen at 6 to 8 weeks of age, and then follow for a year or longer. And what we noticed first, which the field had already known, and but we just wanted to revalidate, was that under normal conditions, bile duct epithelia, marked here in pink, this is KRT19, do not become hepatocytes. And if you mark only hepatocytes but not bile duct cells, there's no change in the unlabeled population, suggesting that everybody stays in their lane. Bile ducts stay bile ducts, hepatocytes stay hepatocytes, and this is sort of known in the field. So we then labeled zone 1 populations using glutaminase, and this population actually, interestingly, shrank over time. And I'm not saying that the cells shrink, but the actual, the zonal thickness shrinks over time. And suggesting that another population is taking over the zone 1 region. And we quantified this by looking at the tomato area based on image analysis. And this is schematized here. This result had also been seen by another group using a different Cree line, so we knew that this was likely to be correct. But we really wanted to make sure by performing the orthogonal tracing using a CYP1A2 zone 2 and zone 3 labeling approach around the central vein. And we got the consistent results which show that this population expands over time to cover more of zone 1 as seen here. And I'll say that this is a very slow repopulation. This is not like intestinal regeneration where over 1 week the whole population is covered. This is a long-term, slow homeostatic turnover. So zone 2 and zone 3 contain something of interest. And it could be that this is consistent with the axon 2 result, showing that the stuff right around zone 3 is repopulating. So we did a similar kind of tracing using glutamine synthetase, which is a very elegant line that only labels right around zone 3. And we found that nothing really changes. So over time, this basically stays the same, suggesting that there's a self-renewing population right around the central vein, potentially fed by Wnt signaling. To kind of reaffirm this, we use the opposite labeling line using arginase 1.1 Cree, and we again saw that this population didn't change much over time. Although we did notice that there's some encroachment of cells into this domain over time, over a very long period of time. So it could be that something over here is taking over if you give it enough time. All right, now to get more at zone 3, we have a line that spares zone 3, as you can see here. And tracing this line suggests that zone 3 is also contracting over time and encroaching upon the central vein adjacent hepatocytes. So these results together suggest that zone 1 and zone 3 are getting smaller over the course of a year, and that maybe another population is taking their place. So this is just to show that we've done the tracing now for 1 year and 8 months in another cohort of mice. And we see that the glutaminase line really shrinks a little bit more, but does not completely go away. The CYP1A2 line expands more, but does not completely take over the zone 1 population. And furthermore, arginase 1.1, which spares that zone 1 population, does get smaller, and the zone 3 population does get smaller and smaller. And I'm showing you the close-up views here, where you can see maybe some encroachment of the population around zone 1, but not complete takeover. And this is zone 3, showing that there's some encroachment from outside of this pericentral adjacent population. These are details, but they're important because it shows that there are independent populations of hepatocytes that are, replenishing each zone. So what is the population that's replenishing under these normal homeostatic conditions? And we've done an EDU trace that is in line with many other EDU tracing experiments done by several other groups, showing that in the midlobular population, there's the most EDU positive cells between zone 1 and zone 3. So the distribution of EDU positive cells is greatest in the midlobular population. And we used a midlobular actually not a great midlobular line, but one that does label a higher percentage of cells within zone 2. And we find that this population labeled by HAMP2 expands over time. We think that together, this data shows that zone 3 and zone 1 contract, zone 2 expands, and this is in part due to an increased rate of cell proliferation within zone 2. And we and others have looked at cell death and clearance. I know that's going to be a factor, but apoptosis is very hard to detect in the normal homeostatic conditions. So we don't know if it's due to apoptosis. We've also done the injury assays, which I'm not going to tell you about today, but this is exactly what you'd expect. It's like a planaria. If you cut off the head, the tail and the body grows to recover. If you cut off the tail, the head and the body recovers the cells. This is exactly what is happening in the lobule. The plasticity of the cells is amazing. And I have to say, in the same issue of Science, Bin Zhou's group performed an orthogonal tracing approach and found the exact same results as we did, showing that zone 2 is where predominance of the repopulating cells are appearing, at least under normal homeostatic conditions. And also Holger Willingbrun's group about a year ago had shown that the zone 2 had the most proliferative cells. So these are corroborating studies that show us that zone 2 is likely to be the most important population for maintenance of liver tissues. So we're running out of time a little bit, but I'll tell you what we know about the mechanistic basis now. We and others have done single cell sequencing, which has clearly delineated zone 1, zone 2, and zone 3. The results are expected as according to other studies and what we've found. We've also looked at very excellent data from the Itzkovitz group, their published data in Nature Metabolism, showing that there are genes that are specifically enriched in zone 2 or specifically downregulated in zone 2. So we focused on this collection of approximately 200 genes, and we again went back to our CRISPR screening approach to ask which ones of these 200 genes are more or less important for hepatic regeneration. And we did both knockout and activation screening, and the two genes that kind of jumped out because they were a little bit obvious were cyclin D1 and HAMP2. HAMP2 is the gene that we use to trace zone 2. It's involved in iron metabolism. Its name is also hepcidin. And we haven't done much on the biology of that yet, but cyclin D1 is an obvious thing to look at. And so when we and others have looked at cyclin D1 expression in 6 to 8 week old mice, there's a very exquisite midlobular expression of cyclin D1. And this is known, but not really paid attention to. You can see that there's this amazing sort of cyclin D1 stripe down the middle of the lobule. And so we did the obvious experiments, which are to chemically perturb or ablate cyclin D1, and of course, proliferation goes away using Ki67 and EDU tracing. We also deleted cyclin D1, and this showed reduction in proliferation, as you would expect. We then asked, well, what's upstream of cyclin D1? And a common culprit is, of course, mTOR, known to be a driver of cyclin D1 expression. And mTOR inhibition using a mTORC1 inhibitor, Inc128, also suppresses proliferation, especially in the midlobular region where proliferation occurs the most. And so then the question is, what is upstream of mTOR in this cascade? And a important gene that we've been looking at is a gene called IGF-BP2. And I didn't show you this line before because we had just generated this new Cre-ER line. And it's really cool because this gene, when you knock in Cre-ER into this gene, it's one of the only zone 2 related genes. And it's got this really nice zone 2 expression pattern, much better than HAMP2, and very similar to cyclin D1. So we're now using this to study the biology of zone 2 under various conditions. But this suggests that this gene could be important in zone 2 homeostasis. It's a complicated gene. It's a large family of IGF interacting proteins. It's a secreted factor, which is thought to either stabilize or perturb the stability of IGF ligands, integrates or interacts with integrins, and overall has a pro-cancer, pro-oncogenic growth factor. And so we overexpress this gene or overexpress this gene using hydrodynamic transfection. This causes a modest increase in PI3 kinase mTOR activation when you evaluate this on the whole liver level. We collaborated with Cliff Rosen's lab from the University of Maine. He had been working on IGF-BP2 knockout mice, and when we knock out the mice, what we see is reduction in IGF-BP2, a reduction in cyclin D1, and a pretty dramatic reduction in proliferation. And you can see that in the Western blots here. So it's very clear that IGF-BP2 is an important factor within zone 2 along with cyclin D1. And so to summarize here, we think that these studies, along with many other corroborating studies, are really showing that hepatocytes in different zones are different from a regenerative medicine standpoint, not just from a metabolic standpoint. We think that mid-lobular populations are preferentially repopulating. And this is something that we're really interested in sort of digging deeper on in different animals and humans at different ages. We really need to understand how widespread this phenomenon is. And I think during injury, there's a lot of plasticity. Many populations regenerate depending on the location of injury. But zone 2 makes sense because it's a little bit sheltered from the edges where damage normally occurs. And we think that this sort of classic growth pathway is a driver of zone 2 growth. And moreover, we think that IGF-BP2 and HAMP1 and 2, which are clearly important disease-related biomarkers, could have a functional important impact on the cell biology of the liver during chronic liver disease. So a lot to study here. I want to thank the people in the lab who've done the studies. Joyce really has spearheaded the CRISPR screening approaches, and she's done a great job with that. Yonglong Wei has taken on the heroic effort of making all the Cree lines. Our mouse core has been instrumental in developing the lines. Min Zhu has done the work for cirrhosis sequencing, and Yu Shanlin is going to do the work on zone 2 and moving forward. So with that, I'll be happy to take any questions and to discuss anything that you'd like.
So there's a question in the chat from Tom Wiki. I will read here. What is the metabolic function of the zone 2 cells that expand into zone 1 and zone 3? Do they acquire Z1 or zone 3 metabolic function, or do they retain Z2 metabolism?
Yeah, that's a great question. We don't quite know. I think so when the zone 2 cells are proliferating into zone 1 and 3, they become zone 1 and zone 3. So they take on the gene expression of the zone that they're in. So it's clearly some kind of a signaling gradient that defines the cell type based on location. And then the other part of it, zone 2 has some metabolic function. There's still hepatocytes with metabolic function, but my impression is that their metabolic gene expression is reduced in comparison to zone 1 and zone 3. And that's what may also allow them to be more proliferative than zone 1 and zone 3. And there's this sort of emerging principle that hepatocytes with reduced metabolic gene expression have higher levels of cell cycle gene expression that may poise them to be regenerative populations under injury conditions.
Yeah, okay, DJ, so you can.
Hey, Hao.
Hey, DJ.
Okay. All right. That's a very nice talk. I have a quick question about the different zones under partial hepatectomy. And so, so if you cut 2/3 of the liver, would all 3 zones proliferate? I would assume, right?
Yeah, they do. So So partial hepatectomy is the classic injury model, of course. You're getting rid of 70% of the liver, and it really recruits all hepatocytes and other non-parenchymal cells to proliferate in response. But, however, it occurs in a wave from zone 1 to zone 2, and lastly, zone 3 probably proliferates the least. And that was nicely elucidated in Bin Zhou's paper that was in parallel with ours. But most hepatocytes will regenerate in that context. And I would argue, DJ, that I love hepatectomy, but we study it too much. And we think of that as regeneration assay number 1. But I think how many of us are getting hepatectomies? We really should be studying modest injuries like alcohol, fatty liver disease, to understand the homeostasis in those settings. And I predict that maybe the less differentiated mid-lobular populations may be important in those contexts.
Yeah, and also actually related part, I understand, I totally agree with you about partial hepatectomy, but still such a miracle phenomenon in biology. So what are the populations under partial hepatectomy that actually replenish the bile duct cells, right? So eventually the recovered liver have to be made of normal lobular structures with portal veins and central veins with all of that. So what is the cell type responsible for generating those epithelial cells?
Yeah, I think the idea is that it's bile duct epithelial cells that give rise to bile duct epithelial cells. But I would say, man, that's just has not been studied very much.
I thought that's an obvious question, right? I mean, I always wondered about it.
And whether or not there's heterogeneity in those bile duct cells, certainly there must be. I just don't think we even have 1 or 2 great bile duct cree-lines to kind of investigate that. Mm-hmm. So I think it's just not studied that much.
Okay, thank you.
Yeah, Jiang has a question. Yeah, Jiang.
Hey, Hao. I'm curious to know, how do you explain the renosis, observation given your findings?
Which one?
Renosis, they find the axin.
Oh yeah, yeah. Yeah, I think that's I think what has happened is that there's a little bit of a difference in that Cree model where this is just, this is what I think. And so the observations that have been made is that when you make a Cree using an axin 2 back transgenic, you no longer see the proliferation from axin 2 positive cells. And also our GS line and LGR5, many other lines in that population show no expansion. The model that they used in the Nature paper was one where the Cree was integrated into exon 1 of axin 2. And so many people in the field believe that maybe one allele of axin 2 was ablated. And therefore, axin 2 is a tumor suppressor, and that modest reduction in dose has an impact on the proliferative response.
Okay.
So it's a lesson, I mean, for all of us, that in the liver, where there's relatively smaller differences between different populations, you really have to be careful about comparing several Crees to make sure that there's no artifacts that can't be explained.
Yeah, okay, so I have a question. So for the IGF-BP2, it's a secreted factor, but it seems it's also expressed in zone 2, also acts on zone 2. Is that what it is, like paracrine autocrine?
Yeah, maybe. Yeah, I mean, so IGF-BP2 is most famously known to be a bone-related factor. So it's secreted from the liver, and it has effects long distance on the bone healing, bone regeneration. And so it's definitely occurring on a long endocrine level, but I think it's also known to act in the intracellular or intercellular spaces in a paracrine fashion. So it could be that the concentration is highest in zone 2, but I'm certain that it's affecting all of the hepatocytes. So maybe it's sort of consolidating that proliferative stuff in the middle and maybe trickling down into zone 1 and zone 3. We don't know yet.
Any more questions? If not, we will thank Hao-Zhu again for a wonderful talk.
Thank you so much for inviting me. Appreciate it.